Thank you so much, Charlie. So we have an exciting talk pair today. Sandra B, Andersen, and Camille Simonet will be talking about harnessing the social lives of microbes to improve human health. And so really thinking about how do microbes talk to each other and how do we think about that to do new things with applied value. And Sandra is going to talk first. Sandra is an evolutionary biologist from the University of Copenhagen in the Center for Evolutionary Hologenomics, where I also have a part-time being. She's worked on ants and their microbes, cystic fibrosis infections, and is now working a lot on H. pylori, but all with an ecological evolutionary sort of framework with humans somewhere in the ambit. Camille is a PhD student at the University of Edinburgh and studies microbial dynamics and social interactions in complex communities. She's previously developed methods to assess the presence and forms of competition in the gut microbiome and to predict its evolution, which is really wonderful work, thinking about what's happening in this black bag, it's not quite a black box, and how we predict it without seeing it all. And Camille's right now working on deriving a microbiome health index using ecoevolutionary rationale to predict what's a signature of dysbiosis. And so how do we bring ecology and evolution and guts and feces all into this medical and applied world? And Sandra and Camille, as I understand it, have never met in person, but Camille had a really exciting recent paper that Sandra was invited to respond to. And so they met through this old sort of Renaissance era discourse in the print pages. And so it's exciting to have their paired talks today. And so I think Sandra will start us off. And so without further ado, would you like to shift?
Thank you so much for the introduction and the invitation. So Camille and I will present our work on using omics data to establish the importance of social interactions in our microbiomes. So in my case, a relatively simple one species community, while Camille's work focus on high diversity microbiomes. And this hopefully should bring us to a point where we can discuss the potential to manipulate these interactions for health benefits. So the microbes that are found in and on us can have a profound impact on our health. Historically, we've of course mostly focused on the pathogens and parasites that make us sick, but we also know now that many microbes are beneficial. And much of symbiosis and microbiome research has focused on identifying these good and bad microbes, but it's not always that simple. So we know that the environment and the context can affect the outcome of an association, and part of that context is of course the interactions with other organisms. So when we think of an organism like a charismatic animal like a lion, it's easy to envision these different types of interactions. We can have cooperation within a species, for example, to gain access to a resource. We can have competition within a species, over mating, for example, and we can have competition between species for a resource. And we can easily see how these are an important part of both the species and the larger ecosystem that it's a part of. But microbes also have more complex social lives than they're usually given credit for. They can interact within a population with other species of microbes, and if they're sitting in a host, with that host. And because of these different interactions, an evolutionary medicine approach is key to understand our microbes. So microbes can cooperate by producing compounds that are not only beneficial to themselves, but also to their neighbors. So this could be the production of a protective biofilm, the breakdown of antibiotics in the environment, and the acquisition of nutrition. So overall, this means that a population of cooperators can do better than a population of non-cooperators. But when we mix the two, we can have a situation where the non-producers are able to exploit the efforts of the producers and invade, and then they can cheat on the cooperators. and prosper at their cost. And we know that relatedness within the population is one of the key drivers of cooperation. And this is something that Camille will talk more about. So we have a fairly good understanding of how these interactions take place we can study them quite well in a lab setting. But what happens when they take place in a host? So this is especially important because many of these so-called public goods, exoproducts that the bacteria can produce can actually affect the host. So I've worked with the Pseudomonas aeruginosa, which is a common opportunistic pathogen of immunocompromised individuals. It's found fairly abundantly in the environment, and it's also the star of many social evolution experiments. So we have a pretty good understanding of the social interactions that Pseudomonas can engage in a lab setting. But we know much less about what goes on in natural and clinical populations. So I've used genomics and phenotypic assays to try and get at that. So Pseudomonas is a major pathogen in individuals with cystic fibrosis. So cystic fibrosis is a genetic disease that causes the development of a thick mucus in especially the lungs and also the upper airways. And this gets colonized by environmental bacteria, such as Pseudomonas. And during infection, we see that a lot of mutations accumulate. And this has typically been interpreted as adaptations to the host environment. But what we also see is that a lot of these social traits of Pseudomonas is lost during chronic infection. So this has led us to ask what drives within host selection. As I mentioned, a lot of these mutations are expected to be adaptations to the host environment. So when Pseudomonas change from living, for example, in soil or water, it obviously has a different nutrients available compared to when it's living inside a host. These individuals are also treated with a lot of antibiotics, so obviously there's pressure from antibiotic treatment. And overall, we would expect that evolved lines have a higher fitness in the host than the ancestor if the changes are only caused by adaptations to the host environment. However, we would also expect that social adaptations to interspecific interactions could play a role in some of these changes that we see. And this may counterintuitively lead to that evolved lines actually have a lower fitness in the host than the ancestor. So I have had the good fortune to address this with an amazing collection of cystic fibrosis isolates from the clinic in Copenhagen. So these have been sampled longitudinally from patients, and we have a lot of different clone types. So one of the things that has been a problem in the cystic fibrosis community has been transmission between individuals. And in the Copenhagen clinic, we have also transmissible lines in the older patients, but they've been separated very well in the younger individuals. So here we have a lot of different clone types from the environment colonizing patients. And all of these have been isolated, cultured, and then whole genome sequenced. So we can look for patterns of convergent evolution between these lines and between patients. So what I've looked at is iron uptake in Pseudomonas. So iron is an essential nutrient for the bacteria, but it's also one that the host actively tries to withhold from invading pathogens. And Pseudomonas has a lot of ways to work around this. So when a bacterial cell is starved of iron, all of these different systems can be activated, and then the most efficient one can be upregulated through a feedback mechanism. So I'm going to talk about the pyrovertin system and the Fu system. And these have some important differences. So pyrovertin is a cooperative system, and a siderophore is secreted into the environment that can bind to iron, has a very high affinity to iron, and it can steal it from host carriers. And then the siderophore and the iron is taken up, and the siderophore can be left out again. In contrast, the Fu system is private because this involves direct uptake of iron in the form of heme through a receptor on the cell surface. So the pyridine system is cooperative because once pyridine is released, the siderophore is released into the environment, it's available to all other cells that have the receptor, whereas this is private because uptake is direct. So from both studies of the Copenhagen isolates, but also from other clinics, we know that a cooperative pyridine production is often lost over time in infections, but not always. And on the other hand, this private heme uptake system is often upregulated late in infection. And so this has been interpreted as a change, as an adaptation to the host environment that is changing, and the iron availability changing during infection. And we, of course, wanted to test whether social interactions could play a role instead. So pyridine is nice to work with because it's fluorescent green and easy to measure. And even better, we understand the genetics behind it quite well. So we have an outer membrane protein, and the siderophore here is released, binds to iron, steals it from a host carrier. It's taken up by the receptor, and this uptake then has a positive feedback loop so that more of the receptor is produced and more of the siderophore is produced. And this site of the receptor is one of the most variable regions in the Pseudomonas genome, and so is actually the whole pyridine system. So it's been under strong selection, or it is an ongoing selection for diversity, sorry. So what do we see? First of all, we see that the proportion of isolates that are non-producers increase during infection. So the isolates stop producing pyridine with time. And then we looked at what are the mutations that causes these changes. And we find a non-random distribution of mutations. So most of the mutations are located in a tiny gene that codes for a sigma factor that controls the whole production of pyridine. So this is the most efficient way to knock out production of pyridine in one step. More surprisingly, we also find a lot of mutations in the receptor itself. So these are mutations that cause lack of uptake of pyridine. And these are only found in isolates that have stopped cooperating and producing pyridine themselves. So we want to figure out whether these patterns of mutations are caused by an adaptation to the lung, so that pyridine is simply no longer needed because there are better ways to get iron, or if it's cheating that drives these patterns. So if it's just an adaptation to the lung environment, we might expect that both the loss of the production of pyridine and the receptor itself happens at the same time. Whereas if cheating is the driver, we would expect to see a maintenance of receptor function when cheating is possible, so that you have a functional receptor that can take up the pyridine that other cells are producing. And only if pyridine is lost from the social environment, you would see a loss of receptor function. And what we find is the support for the latter case. So in the isolates that are not producing pyridine anymore, but are still in a social environment where other isolates are producing pyridine, we never see mutations in the genes that code for the receptor, the sigma factor of the receptor. we only see mutations in the receptor genes once pyroverdin is lost from the social environment. So that means when cheats can cheat, they keep their receptor, and when they can no longer cheat, they lose the receptor. So here we followed clonal lineages of non-producing potential cheats, and we can see they maintain their, they don't get any mutations in the receptor for up to 5 years, as long as they have other bacteria to cheat on, whereas they quickly accumulate mutations in the receptor if there's no one to cheat on. And we think there's so strong selection on losing the receptor if there's no one to cheat on, because the receptor can be used as a gateway by phages and phage-like toxins. So if you can't use it for cheating, then there should be a strong selection to lose it. So is this loss of cooperation just simply game over? Or what happens next? Well, what we see is changes in the private uptake system. So we have this heme uptake system that is much more simple than the pyroverdin system. We have a receptor gene and a couple of genes related to transport of heme into the cells. And what we find is a heavy accumulation of mutations in this tiny promoter region that causes an upregulation of the food system, so it can take up heme at a high rate. Again, we try and follow clonal lineages over time, and we see that for clonal lineages where there's still pyroverdin in the environment, they don't upregulate this food system. If there's no pyroverdin available in the environment, we see an accumulation of upregulation of this private heme uptake system. So basically, we see a privatization of iron uptake when cooperation is lost. So just to sum up a bit, we have a wild type bacteria colonizing patients, and we did some phenotypic assays that I didn't really talk about here. But what we see is often cooperation is actually upregulated in the beginning of infection. And if cooperation is upregulated, we're more likely to see cheats invading, which makes sense because it's easier, cheaper to cheat, better to cheat if there's even more cooperation going on. If cooperation is lost from the environment, well, then we have a privatization of iron uptake, and this doesn't really happen if we still have cooperation. So what's the next step to actually use this clinically? One thing that has been addressed is this development of antivirulence treatments. So if we can decouple bacterial fitness and production of virulence factors, there should be little selection for resistance. This is not always likely to be the case, so that we can disarm without harming bacteria. But it is also predicted that we can target corporate public good virulence factors, and that cells will have a harder time developing resistance to such drugs, because there is this inherent cost of production and risk of cheat invasion. And there has actually been some very nice studies using a quencher of the siderophore pyroverdin, both in vitro and in situ in patients. So what we see here is that bacterial cultures are treated with the compound gallium, which is a metal that binds to the siderophores instead of iron. So it's basically blocking the site that iron otherwise would have used. And the bacteria can't use gallium, so this means that they have a reduced iron uptake. And this is a measure of the resistance that the bacteria develop to gallium treatment, and it isn't very high compared to treatment with an antibiotic. So in vitro experiments and in vivo experiments with mice were very promising, and this has actually also already been tested in a clinical trial with cystic fibrosis patients that were treated with IV gallium for 5 days, And then the lung capacity was measured. And we can see even a month after treatment, they still have a better lung capacity than they had to begin with. So I think this is showing promise, and it's quite interesting and potentially a type of drugs that can be developed and used in combination with standard antibiotics, not as a complete alternative. This was work I've done in Ashley Griffin's lab in Oxford, Søren Molin's lab at the Technical University in Denmark, working with the cystic fibrosis isolates of Helle Krogh Johansen, and in close collaboration with Melanie Gould and Rasmus. And currently we're trying to do to look at some of these patterns in a different organism, namely Helicobacter pylori, in my newly started lab in Copenhagen. So whereas Pseudomonas is an opportunistic pathogen, Helicobacter has a long history of coevolution with humans. And when we find them living in the stomach, they are the dominant bacteria. They were discovered because they have very high costs. They can risk developing peptic ulcer disease and gastric malignancies, but they've also been found to have, there have been benefits found for an infection, as it can protect against asthma and allergies. And what we're trying to do is scale, look at these different levels of interactions between bacteria and a host. So we're trying to look at the cooperation and conflict within the community of Helicobacter, how it interacts with the diverse gut microbiome through the host immune system, and how it affects susceptibility to other parasites. So both of these, the Pseudomonas work and what I'm doing now with Helicobacter, are examples where we really have one dominant species. And in the Pseudomonas work, we use single isolates that have been cultured. But what happens when we have more complex environments, and what sort of tools can we use to address that? So with that, I will give the floor to Camille.
Nice. Thanks, Sandra. I'm going to show that. Right, is my screen shared right now? Yep, nice. Okay, so yeah, Sandra's work shows how cooperative interactions can drive change of functions in natural microbial population, and how we can use omics approaches to follow this. And this idea of manipulating cooperative interactions in microbes can be used to design drugs, but also more generally to manipulate a living community for it to perform a certain function or to restore a certain composition, for example, restoring a healthy microbiome. But we're still quite far from such practical applications of our understanding of microbial social interaction, especially in the context of such complex communities. And as a step toward this, I'm working on developing metagenomics approaches to study cooperation in the context of complex communities with a particular focus on human gut microbiome. And regarding, oh, I cannot change my slide right now. There. Regarding a word on the microbiome and health, we know that microbiome diversity composition is correlated with a lot of health and disease parameters. Leading to this idea, I was saying that we could restore microbiome health using, for example, probiotics. But we're still far from a systematic clinical approach. And sometimes disease are associated with a loss of a diversity, typically in Crohn's disease or obesity, but sometimes, in fact, it is also associated with an increase in diversity, for example, in colorectal cancer. In some cases, specific taxa play an important role, but there are also cases where taxa may be very abundant, but contribute little to the metabolic network. And some more detailed studies of metabolic pathway and enrichment in certain metabolic pathway and genes. Expressions points to some specific mechanisms, but these conclusions are very often disease specific. So all in all, it is still a bit difficult to define what would you include in your prebiotics to achieve a clinical success. And an evolutionary view here can help bringing a more unifying principles by making general predictions that rest upon more by resting upon evolutionary principles that apply more broadly. And within this, my global question is, how can evolutionary theory and social evolution theory help us predict the microbiome functions and the signature of dysbiosis? So one such general prediction coming from social evolution theory regards the importance of population genetic structure. So I'll keep it quite shallow here, but the idea is that people or bacteria in a population can be assorted differently. You can have genotypes that are assorted randomly, so cooperators will randomly interact with either other cooperator or with non-cooperators. And this situation of low relatedness is to be contrasted with a situation of high relatedness, which can result from spatial assortment or from the choice of people you interact with. But in this situation of high relatedness, interactions occur between genetically similar individuals. So if you link genotypes to phenotypes, interactions occur between cooperative individuals. And theoretical work predicts that this should lead to the evolution of cooperation. So this is a simplified version, of course, but the broad prediction is that all else being equal, increased relatedness should favor the evolution of cooperation. And in fact, this is a leading explanation in the evolution of cooperation across various organisms. And only to cite one example, there have been, for example, comparative analysis, especially this one that includes hundreds of bird species, looking at the effect of promiscuity, which would affect this relatedness in the population. And in this study, they showed how high levels of promiscuity, which leads to low relatedness, leads to the evolution of non-cooperative breeding systems, while low levels of promiscuity, which leads to high relatedness, leads to the evolution of cooperative breeding systems. And that was a prediction that was true across quite a broad diversity of bird species. Then one can think, well, in microbes, things are quite different. Microbes are small organisms. They do not look like birds. But in fact, you can manipulate relatedness in the labs, and experiments have repeatedly shown how initiating a population with cooperator and non-cooperator and manipulating relatedness, experiments have shown how high relatedness leads to the increase of the frequency of cooperator in the population. So again, supporting the hypothesis that relatedness favors the evolution of cooperation. But in this case, we're looking at very simplified systems, and most of these studies come from a couple of model organisms. So my purpose here was to ask what is the general predictive power of relatedness, so this idea of looking across a broad diversity of species, while looking at microbes living in natural complex microbial communities. So kind of bringing these two approaches here together. And to do this, I've done a comparative analysis looking at a human gut microbiome. So to give you an overview here, as a real-world data, what I'm looking at is human gut metagenomes that come from the Human Microbiome projects. So from this bag of sequencing reads, what we can do is that then is identify the species that are in a human host and use the patterns of diversity in the sequencing reads to obtain measurements of relatedness, and then do some genomic analysis to assess the cooperative potentials of the different species that we find in this host. And then plug this into a comparative analysis that asks how does the variation of relatedness across the phylogeny of these microbes explains the variation in cooperation. So the step one is to measure relatedness. So again, I'll keep it rather shallow here, but I'm happy to discuss this in more details. The idea is to start with healthy hosts, and from each stool metagenomes from this host, identify the species and compare the metagenomic data across hosts to identify the core genome of this species. And then for each genomic site at the position of this core genome, as use tools, we use these tools called MIDAS, but there are plenty of bioinformatic tools in development at the moment that allow you to do that. And that allows us to identify SNPs variation in metagenomics data and estimate allele frequency at each genomic site. And this allowed us to derive measures of within-host diversity within each host, and then looking across all hosts, obtain a measurement of across-host diversity. Now, contrasting these two scales of diversity within host versus across host is what allowed us to compute measurement of relatedness. So each species present within a given host gives us a point estimate of the relatedness for that species. And comparing the measures obtained from different hosts, we can obtain an average measure of relatedness for a given species. So of course, all species are not found in all hosts, so we have different level of certainty in the estimate of relatedness of each species. But what we did is taking the average across this point estimate as a measure of the global average relatedness experienced by a given species of the human gut. And we've obtained measurements like this for about 100 species common in the human gut. And so they're plotted here with the species here on the y-axis and the relatedness here on the x-axis, where the mean relatedness will always range to 0 from a maximum to 1, which is the maximum relatedness, which corresponds to a full assortment. And what you can see is that most of the species in the gut human microbiome have a relatedness higher than 0.5, which means that two interacting bacteria in the microbiome are as genetically related as would be two siblings in classic sexually reproducing individuals. But we do observe variation from 0 to 1, which means that we have a great deal of variation to then assess how does this variation explains variation in cooperative behaviors. So that brings me to the second step, which is to measure sociality for these different species. And for this, we developed computational approaches to do this from the genome content of the species. So this starts from one observation, that is that whenever we're looking at a classic bacterial cooperative behavior, for example, a biofilm where bacteria secrete an extracellular polymer matrix, or quorum sensing where bacteria secretes molecules to communicate, or in antibiotic resistance where a bacteria will secrete antibiotic enzymes, enzymes that degrade the antibiotic. All of these cooperative behavior involves the secretion of products. So a generalization is that public goods in general are secreted products. And that is quite a handy observation, because we can predict whether or not a molecule will be secreted or not based on its sequence motifs. So we used the representative genomes of the different species and used tools. I used this tool called P4V to predict how many of their protein coding genes are coding for genes that will be secreted, and use these as a measure of potential cooperativity, where we expect species that code for more genes that are secreted in the environment to be species that are involved in more cooperative behavior. Now, we also wanted to obtain measures of cooperativity that target more specifically different forms of cooperation. There are well-known cooperative behavior in bacteria. I've cited biofilms. Sandra talked about siderophores. Other well-known are quorum sensing and antibiotic resistance and a degrading enzyme. And we can also look at secretion systems as a more nuanced, a distinct category from the secreted molecules themselves. And so to obtain measures that target more specifically these different big forms of cooperation, I've used gene ontology annotations, which have the nice feature to be organized in such a way that you will have parent and child terms like this. So this annotation system will have, for example, a tag for biofilm information, and it will have child terms that relate to these biofilm informations. And these terms themselves will have child terms. And so that facilitates the assemblage of collating a list of annotating terms that will accurately capture a given cooperative behavior. So what I did is that I collated a list like that captures each different forms of cooperation in bacteria, and then annotated with geoterms all of the genes of the representative genomes of the species I was studying. And then intersecting the annotation with this list, you can again obtain a measure of cooperativity, this time by counting the number of genes that are annotated to code for one of these specific terms. And this gave me measurements for 5 more specific forms of cooperation. So the overall structure of the data then looked like this. I have about 100 species included, which covers about 40 genuses. And I have measured this predictor variable that is relatedness, and we're trying to see how along the phylogeny of these microbes it predicts the variation in these different forms of relatedness. And so I've plotted here for each form of cooperation the estimated regression coefficient, and we found that for the categories of siderophores, biofilms, and secretome, we found a positive association between relatedness and the corresponding gene categories. And although for the 3 other categories we did not find a significant association, when we look across all forms of cooperation, accounting for the uncertainty with which we estimate each effect, we find an overall global significant association between relatedness and cooperation across this broad diversity of microbes. So coming back to the overall goal of understanding the dynamics of complex communities and the possibility of engineering microbial communities, there are plenty of contexts where this could be useful. In human health, for example, by engineering fecal transplant or by restoring microbiomes, but also in food production to enhance food production, or in bioremediation. In all of these possible applications, one thing that is important to advance toward a real microbial community engineering science is to identify mechanisms that are of general importance, which can help us understand how microbial dynamics across various systems with different degrees of complexity work. And our results here suggest that relatedness is one such mechanism. It predicts the evolution of cooperative behavior across a broad diversity of species with a discernible signal in real-world data of a highly complex microbial community. Now, relatedness itself will likely be affected by a range of ecological factors, including group size, transmission ecologies, the host immune system. But these are very multidimensional data, and it's actually quite still poorly understood how these different factors collectively shape relatedness, even less so in microbes. But this gets to the heart of why relatedness is relevant quantity for this kind of broad prediction, because it is a single population genetic parameter which collapses a great deal of complexity of these various aspects of the ecology of different species of microbes. And we've shown here how we can leverage on existing data, metagenomic data, and recent bioinformatic tools that allow to analyze this data in a way to quantify this parameter. And this makes relatedness a potential actionable tool to monitor and manipulate microbial communities. And this brings me to a conclusion on how sociomicrobiology potentially fit within this science of microbiome engineering. Metagenomics is already used to reveal species composition of a microbiome, as well as its metabolic potential, or for example, its antibiotic resistance profile. And here we're bringing in a sociomicrobiology dimension where we can use genomic approaches to evaluate the potential of different species of engaging in different cooperative behavior, and how relatedness might affect that, and potentially we could tweak relatedness to modify these functions. And altogether, these different tools allow us to develop, hopefully, a rational approach for microbiome engineering. So with this, I'd like to thank Luke McNally and Rosalind Allen, who are my supervisor at University of Edinburgh, and Jawed Hadfield, Dan Nursing, Kevin Foster, who all provided valuable feedback on this work. And yeah, and thank you all for your attention.
Thank you so much, Camille, and thank you, Sandra. Those were really exciting talks. As you probably have seen, there's a lot of chatting going on already about them. And so I think we have a lot to say, but what we're gonna do today is that Joe Alcock, who's an emergency room physician and also a very clever thinker in evolutionary medicine, is going to be a discussant and sort of initiate some response to these talks, and then we'll go from there to your questions. And as Meredith points out, please raise your hand if you have something you want to introduce. There's a lot already in the chat, but and so we'll pass to Joe now here. Joe, do you have access?
I think so. Yeah, thank you, Rod. And thanks for a couple of really great presentations by Sandra Andersen and Camille Simonet. Thanks for sharing this really exciting work. I have a ridiculous number of possible questions, and of course, we're not going to get, we're not going to scratch the surface, but that just, I think, lends the importance of this topic and to the models that both of you have discussed here. From a clinician standpoint, I'm interested very much in how we can clinically manipulate these features for health in patients. And it occurs to me that both of these topics focus mostly on what I think are pathogens. Certainly Pseudomonas is a pathogen. And then the work by Dr. Simonet showing that the cooperation features, most of them I think were also associated with pathogenicity, things like quorum sensing and antibiotic degradation and that sort of thing. So one question I think to start off with is, what are the best ways that we can manipulate these features? And does it necessarily follow that inhibiting public goods by these kinds of microbes is going to increase patient health? I can imagine other scenarios in which that might not be the case. So for instance, bacteriocins produced by E. coli that kill off competitor strains, that might increase relatedness and be a measure of cooperation, but that may actually be good possibly for a host. It's not obvious. So that's where I'd like to start that. I'm also curious about what do you think is the effect of antibiotics on these systems in terms of changing cooperation? Because it occurs to me that antibiotics would tend to increase relatedness and might increase cooperation if that has a causal relationship, and might thereby decrease health. So I'll start with those kind of general questions to either one of you.
I can start with a quick comment. I think so obviously my system is definitely pathogens where we want to get rid of them. But if we are looking more broadly, such as in the gut microbiome, I think if we can couple, if we have an idea of which organisms we want to encourage and have there, and we figure out which interactions are important for them, then it can either be, then it's no longer trying to disrupt those interactions, but actually encourage them instead. So I think if we can couple it with the organism and what we want with them, then I think that's a first step.
That's great. It does raise the question, when is a public good a patient bad? And thinking about it in that way might be useful. And a question for Camille. I'm struck by the fact that E. coli in the paper that you recently wrote E. coli was the most related of the microbes that you looked at, and it made me really think about this idea of a pathogen bloom being part of what's going on here. That there may be certain microbes that gain some sort of advantage and are able to increase their population in a way that also is associated with increased relatedness, and that may be something that gives them some sort of advantage. But I also was wondering about whether we think, how does this interact with the immune system? Is it a goal of the immune system to prevent cooperation or to reduce relatedness? What do you think about that?
So yeah, regarding the E. coli having the highest relatedness, I'm not too surprised by this, as in because so these measures of relatedness of course contrast diversity within a host versus the diversity across hosts. And for bacteria that we expect to be massively present and shared easily between hosts, of course you will have the diversity within hosts will resemble that across hosts, and that will lead to very high relatedness. And there is also, of course, various aspects of the ecology of microbes that will lead to variation in relatedness, and we are looking a little bit into that. And you mentioned blooms of bacteria, and like what is traditionally predicted by theory in this area, we found that group size seems to be associated with higher relatedness. So it seems that there is a positive association between, for example, blooms of bacteria and lowered relatedness. So one possibility would be that when bacteria increase in, blooms of bacteria leads the host immune system to favor, for example, one particular strain, which would reduce its within host diversity and leads overall to a high relatedness. So it is very possible that there is an interaction between these dynamics at the species level and the role of the host on shaping this diversity. That being said, the role of the host on the relatedness, or at least the within species diversity in general, is quite hard to assess. I'm not too sure from what we know today. We know that hosts can select certain species, but I'm not sure to what extent a host can select certain specific strain within a given species, which would be an interesting avenue of research, of course.
Yeah, I bet the immune system does target at least those genes that are associated with cooperation, if to the extent that they're also associated with virulence. And I was wondering, this will be my last comment before we open it up more generally, but have you considered looking at cooperation in other microbiomes? So for instance, maybe the vaginal microbiome, where the public good of a lactobacillus to decrease the pH excludes pathogens. That's an area where cooperation among microbes benefits the host.
Yeah, I think that would be obviously a very interesting question, and the method is there, and now applying these methods to other systems is rather straightforward. One thing that is interesting about the gut microbiome is the presence of many species. There are less species present in other, for example, in the vaginal microbiome, so it might be a bit harder to derive more general conclusions, and obviously the number of species that you can include in your analysis is important to have some power to make more general predictions, but I think that would be definitely a relevant question.
Thanks. Other questions?
So it looks like we have a hand raised from Bernie Crespi. Bernie, you can go ahead with your question.
Hi, yeah, I wanted to thank the speakers. It was absolutely wonderful. My question had to do with what I suspect are among the worst of the killers in the microbial world, and that's the bacteria that are involved in sepsis. And I'm just wondering what we know about what's going on in those with regards to cooperation and cheating, and how that might be useful, because just in terms of the death rate and proportion of people affected, that seems like a really important situation.
I know that there has been a bit work on, I believe, staph infections, blood infections, and that there are some of the virulence factors there that are cooperative, that potentially could be targeted here. So there's definitely potential to look at that.
And anything known about their relatedness? Are they just getting a big clonal explosion and they're highly cooperative in those cases, for example? Or
I don't know the data on it. I don't know if it's been done, but I would imagine so, just from like biological point of view, right? That it would be likely that an infection would have been established by one like a small population.
I mean,
But
a lot of
the work has
sepsis.
been done.
A lot of sepsis is polymicrobial.
Oh, really?
Well, at least certain. I mean, I'm sure we can find examples where there's a dominant, bacteremic strain, and there of course is sepsis in which we can never identify the causative organism. So I think that there's variation there.
That's an interesting contrast, actually, because the fact that sepsis is polymicrobial, that it would be interesting to contrast between species diversity. Like it's polymicrobial, but each species might be of a very low relatedness. Yeah, that could have implication of having sepsis of high or low within species diversity, I guess.
Great, thank you for that. So there was a lot of great questions in the chat, which I know y'all have been diligently answering, but there's a couple that I wanted to bring up to have answered live as well. Amrita Bhattacharya, I would have her ask verbally. It looks like she's had to tune out, but she had some great questions about what caused the initial loss of cooperation and whether loss of cooperation was more often what happened or whether long-term maintenance of the cooperation was more common. So interested to hear your thoughts on that.
Yeah, so definitely loss of cooperation is the most common. But what we think is going on is that in some patients, there's an initial increase in cooperation. And this increase could be driven by availability of iron, so that it's beneficial to produce more siderophores. But it could also be, for example, a competition with staph that we often see early in infection, co-infections with staph. And so basically, because you can see it as a kind of interspecies iron piracy, because when Pseudomonas produce siderophores that bind to iron, they're only available to Pseudomonas with the right receptor. So this could be a way to limit competition with other bacteria. And then when we have this increased cooperation levels of cooperation, then it's also easier for cheaters to invade. So this is what we think is going on, but it's not always the case that the cooperators invade. But yeah, that we also had some talk in the chat about what actually are the levels that the scale that bacteria are interacting at in the lung, because obviously this mucus is quite thick and there's probably little diffusion of siderophores within the lung, but there is some turnover and mixing, but this is very difficult to quantify. So actually getting at the dynamics is tricky.
Great, thank you so much for that. So we probably have time for one quick question. If anybody has any burning last questions, feel free to raise your hand now. Looks like we have a question from James Gurney. James, go ahead with your question.
Hi, how are we doing? This is a question for Camille, I guess. I assume it was a Bayesian analysis you did on your sort of different traits. Why do you think QS wasn't like had a I don't want to say significant. Why was this coefficient not positive in this case? What do you think the rationale there is?
It's very surprising, right? Because QS is such a widely
recognized. It's the social
thing, and there are a lot of quorum sensing based drugs being developed, etc. My hypothesis is that quorum sensing mechanisms are themselves very diverse, and it would be interesting to look into more details, like have an even more refined measure of quorum sensing to be able to target a specific type of quorum sensing. I know as well that analysis a bit similar to what Sandra has been looking at with siderophores. There are analyses being similar with quorum sensing. And so in a sidereal system, it tends to lead to the upregulation of the private systems. With quorum sensing, it seems to lead to a diversification of the specificity of quorum sensing with like more specific receptors, etc., diversification of receptors. So I think quorum sensing is so important and is coordinating so many aspects of bacteria expression, gene expression regulation that it might be a little bit more complicated than other forms of cooperation. So it would require more detailed investigations of the specificities of what we're targeting with this way of annotating, at least. One other aspect is that I think compared to the other measures, there might be also a lack of statistical power because compared to the other measures, we captured less genes coding for quorum sensing in these particular set of bacteria. So that would be something too that could be looked into more detail as well.
Okay, fantastic. We're at the top of the hour. And so we have to close down. Unfortunately, there's so many great questions still to ask and to consider. Sandra, thank you for a great talk. Camille, thank you for a second great talk. Great idea, Sandra, to have the two of you go together. I think that worked out really well. And Joe, thank you for your perspectives at the end and for leading us into the questions. I think this was a real success. So thank you guys very much for being a part of this. Before we tune out, I want to share with you the upcoming Club EvMeds. Hopefully I'll do this right. Hopefully you can see the Club EvMed website now. I hope you've been to the Club EvMed website. Here, of course, is the great talk we just heard today. Next week, we're going to be hearing from Joe Henrich at Harvard. He's going to be talking about his book, and it should be a very interesting discussion. Hope you can make it. After that, we have Zane Thayer from Dartmouth talking about the causes and consequences of fear in of childbirth. After that, we have Beth Archie and Chelsea Weibel talking about their work recently published in PNAS that I think you'll find very interesting, especially if you're interested in the whole world of life history strategies and predictive adaptive responses. And then one thing I really want to call your attention to is we have a postdoc spotlight coming up. Please go to our website and click on this link and you can fill that out for yourself if you would like to be one of those postdocs or to nominate somebody. So please follow that. And then later in the semester, really after the semester, we have a student spotlight. So if you're a PhD student and you'd like to present your work at Club EvMed, we're going to have a series of 3 speakers for postdocs and 3 for graduate students on those 2 days. And nominate someone as well. You can do that. Okay, with that, I'm going to close down here. Thank you guys very much for tuning in, joining us today. And thanks again to the speakers for great talks and to everyone for these great questions. So thanks very much, guys.